everyone. Myself Ruchita Karmakar, and my presenting a talk on microRNAs and the epigenetic modifications in the glioblastoma and the neuroendocrine tumors.
Good evening, everyone. Good morning, good evening, good afternoon. Have a good day, everyone. This is Narendra Singh, and I'm currently pursuing MSc in biotechnology from Amity University, Kolkata, in India. And me and Ruchita will be speaking together on the topic microRNAs and epigenetic modifications in glioblastoma and neuroendocrine tumor.
So, glioblastoma are also known as the grade 4 astrocytoma, which is a rapidly growing and aggressive brain tumor. It invades the nearby brain tissue and usually do not spread to the distinct organs. The glioblastoma is a devastating brain tumor that could kill within 6 months if it is left untreated. Therefore, it is imperative to seek the changes and immediate treatment by the specialist in the neuro-oncology and the neurosurgery overall. The gene expression in the cells and the tissue very complex organism is precisely controlled and largely dependent on the different conditions such as the development, changes in the environment, diseases, or the drugs. The non-coding RNA molecules have a very specific role in the plethora of the regulatory events, from the controlling the number of copies in bacterial division to X chromosome inactivation in mammals. So basically, the microRNAs constitute a new class of small non-coding endogenous RNAs that basically regulate a gene expression by directly targeting the mRNAs for degradation or translational repression. Their findings added a new dimension to our understanding of complex gene regulatory networks in humans as well as in animals. Because these microRNAs control many aspects of development and are becoming increasingly important. Analysis of microexpression and information as regulation disease such as and diseases immune disorders. Next is on how do these immune checkpoints and inflammatory molecules in glioblastoma targeted by the microRNAs. The immune checkpoints can be modulated by microRNAs and complex network between the gene immune checkpoints. Intuitively, this complex network requires tuning of the checkpoints. The microRNAs, for example, the miR34A and miR130A, same time can directly promote PDL1 and PD1 immune cells expression of the cytokines that in turn induce the expression. The microglia and astrocytes, along with the macrophages, are a part of this glioma microenvironment, and the brain immune system expresses cytokines and are part of this regulatory molecules. Interestingly, there is miR146, which is a negative regulator of astrocyte mediated inflammation and upregulation of this microRNA decreases the expression of its target TRAF6 that is linked to seizure frequency in the glioma patients, suggesting that miR146 could be involved in the epileptogenic focus surrounding the brain tumor. Our next
Sorry
talk
to
is on the invasiveness.
Sorry to interrupt, but Ruchita, slides are not visible actually. Like it's not changing.
The slides are working in my screen. I think it's a problem you have. It's not the problem in her screen sharing.
Yeah, it's okay now. It's
Yeah,
okay.
for
Thank
me
you.
too.
Okay.
Thank you. So our next talk is on invasiveness and the blood-brain barrier treatment that escape the mechanism. So an important mechanism contributing to the treatment failure in the glioblastoma is the tumor invasiveness. The brain is a specific environment made opaque to the outside world, and the molecules that cross the blood-brain barrier. This prevents the efficient targeting of the glioma cells by anti-neoplastic agents unless the blood-brain barrier is severely damaged, as in gliomas. Nevertheless, the glioma can also evade treatment due to its ability to invade the brain, where the blood-brain barrier forms niches in areas that are intact. Invasiveness occurs due to the mesenchymal transition, or the EMT, which is a kind of mechanism by which the cell loses their epithelial phenotype and acquire a mesenchymal phenotype. Several microRNAs are involved in the epithelial mesenchymal transition, or the general entry mechanism. These may include certain microRNAs like the miR-21, miR-146, which may directly or kind of indirectly target the metalloproteinase inhibitor adhesion molecules and other genes which are basically involved in the metastasis and the cell invasion. The glioblastoma is a highly vascularized tumor, and this feature may contribute to the tumor's ability to invade and proliferate, as well as the shielding of the glioma cells behind the blood-brain barrier. The overexpression of the VEGF and the interference of the VEGF is not only the angiogenic mechanism exerted by the microRNAs in the glioma. In fact, the angiogenesis is promoted by miR-93 targeting the integrin beta 8, which is basically involved in the cell-to-cell interaction. And a set of microRNAs can also directly or indirectly fine-tune and promote the angiogenic factors. So there are some other extracellular vesicles, such as the exosomes, which may be involved in the non-pathological situation or as means of the communication between the tumor cells and their environment, and thus may trigger the cells. The next is the therapies for the glioblastoma. The developing a multi-omic strategy should be impressive, significant, and advances in the knowledge of mechanism. In principle, the drug-based biological therapy is sought modification of cell phenotype by molecules, where we can basically block the well-defined paths, maybe due to the inhibition of the enzyme function by activity of certain proteins. The mesenchymal stem cells are an interesting and potentially highly effective way to target the sites of injury or inflammation and tumors for the therapeutic purposes, and the co-culture with the appropriately engineered mesenchymal cells. And it has been shown that functional microRNAs can also be transferred into the neural progenitor cells. Another interesting example of a cooperative treatment of this glioblastoma is provided by a multifunctional delivery system, MSNPs, which is the mesoporous silica nanoparticle that is charged with TMZ molecules, and it is kind of decorated by an anti-miR-221 PNA octa-arginine conjugate that increases the biological effect of the TMZ molecule, which has been attached to the MSNPs, and in a drug-resistant cells. A similar effect was seen in the miR-34A encapsulated in a polyglycerol scaffold. So nanoparticle appears to be a very promising strategy and has been used to deliver not only various drugs, but also microRNAs to the tumor sites. In principle, Nanoparticles should predominate poor systematic stability of the oligonucleotides, facilitating their delivery. Different nanoparticle formulations are available, which are advantages and disadvantages in both the ways. However, they can be designed to target tumor sites and across the blood-brain barrier in the case of the brain tumors. The next slides will be explained by Narendra Singh.
Thank you, Ruchita. About DNA is exposed so that the gene is turned on. Chemical groups can be added or removed from the histones to make the histone more tightly or loosely packed, turning genes on or off. So that is the basic preliminary idea of what exactly is epigenetic and how the changes occur. Next slide, please. Yes. So now comes the application part. How can we actually apply non-coding RNAs and epigenetics, which will eventually control the gene expression? Now, as Ruchita mentioned before, non-coding RNAs are, as the name itself suggests, are a set of RNAs which are smaller than the messenger RNAs, and they do not code for any protein, or rather they do not get translated to form a polypeptide. Now, DNA is used as an instruction for making coding and non-coding RNA. Coding RNA is used to make proteins. Non-coding RNAs, on the other hand, control the gene expression by attaching to coding RNAs along with certain proteins to break down the coding RNA so that it cannot be used to make proteins. Non-coding RNA may also recruit proteins to modify histones to turn the genes on or off. Now, how that happens is a very interesting process, because when a non-coding RNA recruits certain protein, a very common example it's given over here in it's also shown in the diagram. Uh, for example, three non-coding microRNAs, miRNA 29, miRNA 29A, and miRNA 148. The three of them together control the activity of two methyltransferase enzymes, DNMT3A and DNMT3B. Now we know that these enzymes are responsible for transferring the methyl group onto the DNA, and when that happens, it leads to switching off of that particular gene. So when these two methyltransferase enzymes bind to a particular tumor suppressor gene, which means that the role of the gene is to form a protein or a polypeptide, which would eventually lead to we can say that it has tumor suppressing abilities. Now these two methyltransferase enzymes go and bind and transfer the methyl groups over there, which leads to the methylation, and eventually it switches off the particular gene, and it then leads to the formation of cancer, and it is widely studied in glioblastoma as well. And the enzymes are regulated by three microRNAs. In another example, and the previous example is also, I have tried to describe it in the particular diagram. The next example, which is a very, very hot topic right now in epigenetic cancer research, is EZH2. Now, EZH2, it codes for a particular enzyme, protein, which later becomes an enzyme, and which is histone methyltransferase, and it catalyzes the trimethylation of H3 histone, which means the addition of three methyl groups, which would also lead to reduced expression of that particular gene, or I can say heterochromatization of the particular gene. It causes the post-synthetic modification, leading to transcriptional inactivation, as I mentioned before. Now, this is eventually studied in glioblastoma and other tumors. EZH2 is overexpressed and acts as an oncogene with multiple modes of action, including cell invasion, utilizing largely tumor-specific mechanisms. Now, several microRNAs and long non-coding RNAs are integral components of mechanisms that regulate EZH2 expression. As I mentioned before, the enzyme is regulated by several microRNAs and long non-coding RNAs. That's the beauty why we are. That's the reason why we are studying this thing, and that's the beauty of molecular biology. And again, some of the properties have been known, but most of it still remains unknown, and a huge number of papers are coming up, and a very broad research is going on in this field. Next slide, please. So we have seen the problems, and now what are the possible solutions of this problem? Now, firstly, as I mentioned before, EZH2 is coding, it's like a methyl transferase enzyme, and hence if we can knock out this particular gene, it would end all our problems. But that would eventually create further more problems, because it would this particular gene influences the expression of several related genes, and hence this would lead to endless problems. A possible solution here is that uh, we can actually inhibit this particular enzyme, and inhibition of this enzyme would lead to reduced tumor progression, and also as I mentioned before, that these changes are reversible in nature. They are not like genetic changes which would be irreversible. They are reversible in nature, and hence if we inhibit the particular enzyme for a very long period of time, it would lead to the you know decreased survival. But on the other hand, if this inhibition is for a short period of time, it leads to much more survival rates in the animal models. Yes, it has been tested on animal models till now. We haven't reached humans yet, but someday it will be tested on human models as well. The research work is still going on. Now, again, acetylation and deacetylation, as I mentioned in the beginning slide of the epigenetics as well. Acetylation is, totally opposite of that of methylation. Acetylation is carried out by a specific set of enzymes known as HATs, or the histone acetyl transferases. Now, these process of acetylation, it leads to transcriptional activation or euchromatization, which means that the gene would be feasibly transcribed into an mRNA. And deacetylation does the reverse, which means that removal of the acetyl group, it is carried out by a specific set of enzymes known as HDACs, or histone deacetylases. Now, this leads to transcriptional suppression and heterochromatization of the particular gene. Now, in glioblastoma, the expression of HDAC1 and HDAC3 is inversely correlated with the survival of the glioblastoma patients, which means that it is contradicting the original findings. The two enzymes are inversely correlated, though they both have the same function of, removing the acetyl groups. On the other hand, the expression of HDAC4, 5, 6, and 11 is positively correlated with the survival of the glioma patients, which means that if we can actually utilize these particular enzymes in the affected the cells, they effectively remove the acetyl groups and increase the survival rates of the glioma patients. Also, the half of the known microRNAs are hosted in the CPG-rich regions and are thus potential targets for DNA methylation. As I mentioned before, methylation occurs specifically in the regions which are rich in guanine and cytosine, that is the CPG islands. And if CPG islands, it has been also found that half of the known microRNAs are present in the CPG islands, which means that they are targets for DNA methylation as well. Also, the effect of DNA methyl transferase inhibitors, which means that the enzymes the factors which would inhibit this particular enzyme and also the inhibition of methylation. They have been tested on several cancer cells and showing the activation of the microRNA target gene axis, and it has also yielded some positive results. And an example which I would like to mention over here is 5-azacitidine, which is a common drug used in this case. What happens is that 5-azacitidine is an substrate analog of the methyl groups, and what happens is that it inhibits the enzyme activity, and it's a very common drug. It's right now it's used as a very common drug. Next slide. So conclusion and future prospects. So again, the epigenetics is comparatively a very new area of research, and we know very little about this. We are still beginners, and we are still exploring the possibilities, and a huge amount of study is needed to be done, because in my previous slides also I said that there are though the same group of enzymes, the HDACs or deacetylases, they perform the same function, but again the different classes of these of the enzyme had different effects on the patients, which means that our knowledge is still very less, and a lot of contradictory data has also been seen, and we need to study more and more about this thing. So again, although our knowledge of the complex effects and interactions between microRNAs and genome is still incomplete, the potential implications of microRNAs for the diagnosis and prognosis, for the patient stratification, for the personalized therapies were not overlooked. In order to translate the impressive basic knowledge so far gained on microRNAs onto the clinical practice, several issues urgently need to be addressed. Besides the techniques of delivery and targeting, the major problem remains that of understanding the microRNA effects not anymore at the level of single microRNA target interaction, but utilizing a holistic approach to fully appreciate the balance between microRNA and target genes of opposite functions. Now, microRNAs itself, the concept is was not very much familiar some 20 or 30 years back. So we are also trying to study the relation between microRNAs and its related genes, how they are interacting together. And as we move further, like in the coming future, we will definitely have much more data and much more information about it. Thank you.
Thank you very much for your presentation.
And if anybody has any question, you can feel free to ask.